Have you ever heard of single-cell protein, or SCP? SCP refers to the dead, dry cells of microorganisms like yeast, bacteria, fungi, and algae. These cells are rich in protein and can serve as a food or feed supplement, making them a sustainable alternative to conventional protein sources. What makes SCP special is that it contains a high content of protein along with all the essential amino acids our body needs. Some common examples of SCP include: from bacteria, Methylococcus capsulatus and Bacillus subtilis. From yeast, Saccharomyces cerevisiae and Candida utilis. From algae, Spirulina and Chlorella. And from fungi, Aspergillus niger and Fusarium venenatum. Among these, spirulina is one of the most popular and widely accepted foods across the world. Spirulina is a microscopic, one-celled, blue-green algae that thrives in warm freshwater bodies. It contains an astonishing 65 to 70% protein, the highest concentration found in any food, plant, grain, or herb. So, how is spirulina produced? First, we set up a tank or pond. Cement tanks, plastic tubs, or lined ponds with a depth of 1.5 to 2 feet work well. Spirulina needs plenty of sunlight, so we choose an open, sunny space. Next, we prepare the water. It should be clean and non-chlorinated, with a temperature maintained between 30 and 35 degrees Celsius. A nutrient mix, also called culture media, is added. This mix includes sodium bicarbonate, urea or ammonium sulfate, magnesium sulfate, potassium sulfate, common salt, iron sulfate, and other micronutrients. Once the medium is ready, we inoculate it with a starter spirulina culture from a farm or laboratory. For daily care, the water is stirred 3 to 4 times a day and the pH is maintained between 8.5 and 10. The temperature and sunlight are carefully monitored, and the culture is protected from contamination. After about 7 to 15 days, the water turns dark green and thick. This means spirulina is ready to harvest. It is filtered using a fine cloth or sieve, washed thoroughly to remove excess salts, and then dried. Drying can be done in the sun, shade, or using a solar dryer for better quality. Finally, the dried spirulina is ground into powder or pressed into tablets and packed in airtight containers, ready for sale or consumption. Now let's look at the benefits of spirulina. It helps increase metabolism, boosts the immune system, and even reduces anxiety. Spirulina also helps in lowering cholesterol, supporting weight loss, and may even play a role in preventing cancer. In short, single-cell protein, especially spirulina, is a powerful superfood with immense potential to meet the world's growing protein demand in a healthy and sustainable way.
Let's talk about yeast as a source of single-cell protein, or SCP. Yeasts are among the most common SCP producers. Why? Because they are non-toxic, fast-growing, and rich in protein. Some important examples include Saccharomyces cerevisiae, also known as baker's yeast, Candida utilis or torula yeast, Candida tropicalis, and Cluveromyces fragilis. Now, let's look at the characteristics of yeast SCP. The protein content is about 45 to 55% of the dry weight. They are rich in essential amino acids like lysine, threonine, and tryptophan, but limited in sulfur-containing amino acids such as methionine. They also contain nucleic acids, which must be reduced before human consumption, otherwise they can lead to uric acid buildup. Apart from protein, yeast SCP provides B vitamins, minerals, and polysaccharides. What about the substrates used for yeast SCP production? Commonly used sources include molasses from the sugar industry, whey and corn steep liquor from agriculture, and wood hydrolysates. In the past, petrochemical byproducts like paraffins, ethanol, and methanol were also used, but these are now less common due to safety and cost concerns. The production process involves several steps. Step 1 is fermentation. The substrate, such as molasses or whey, is diluted and enriched with nitrogen sources like ammonium salts or urea, along with phosphates, magnesium, and trace elements. The pH is adjusted to around 4.5 to 5, which helps prevent bacterial contamination. Next is sterilization. The medium is sterilized by heat or filtration. Then comes inoculation, where a pure starter culture of yeast is introduced under aseptic conditions. The fermentation is carried out aerobically, at a temperature of 28 to 32 degrees Celsius, with continuous aeration and agitation. This ensures the yeast grows respiratorily, producing more biomass instead of alcohol. As a result, the yeast cells multiply rapidly, converting the substrate into valuable biomass. The next stage is harvesting. The yeast cells are separated from the broth using centrifugation or filtration, and then washed to remove any remaining medium. A critical step is RNA reduction. Yeast naturally contains about 5 to 10% nucleic acids. If consumed in high amounts, this could lead to uric acid buildup and health problems like gout. To solve this, the yeast undergoes heat shock at 60 to 70 degrees Celsius, which activates RNA-degrading enzymes, or it may be treated chemically or enzymatically to lower RNA levels. Finally comes drying and processing. Methods like spray drying, drum drying, or fluidized bed drying convert the biomass into powder. This powder can be packed as nutritional yeast in flakes, tablets, or capsules, or mixed into animal feed. Now, let's see the applications of yeast SCP. For human food, it is sold as nutritional yeast with a cheesy flavor, rich in B vitamins, or as brewer's yeast dietary supplements. In animal feed, it is used for poultry, cattle, and aquaculture feed. Industrially, yeast SCP is a source of enzymes like invertase and zymase, vitamins, and even probiotics. Medically, it is valued as an immunity booster, for supporting gut health, and for reducing cholesterol.